Third, so if you have two different versions of the gene, two different alleles, then one, if there's the dominant allele present, it's going to make the dictate the appearance of the offspring. If the recessive allele is present, it remains in the progeny, although you might not be able to see it because it is masked by the presence of the dominant allele. So all of those F1 plants appeared purple because the purple pigment allele is dominant to the recessive white allele. So when an organism produces its gametes, when it goes from being diploid to being haploid, the gametes can only get one version of a gene. You can only get one copy of each gene in a gamete because they're haploid. In the diploid state, you've got two copies of every gene, so you can mask recessive characteristics. But if you produce gametes that have recessive alleles in them, you can pass on those recessive alleles, and they can be seen in the progeny. So an egg or sperm is only going to get one of the alleles that's present in the parent. Another way of saying this is that during gamete formation or meiosis in animals, the alleles are going to separate, and then you have them coming back together again at fertilization. And this explains why we see that 3-to-1 ratio in the F2 generation. It's a lot easier to see if we come up with some sort of a diagram. We can use a diagram called a Punnett square to represent what's happening. And so we can, what's often done in these situations is we use a capital letter to represent the dominant allele and the lowercase letter to represent the recessive allele. So here we have true breeding parents, true breeding purple flower parents. So they have two copies of the big P allele. True breeding white flowered parents, which have two copies of the small p allele. They produce their gametes, and because they are both homozygous, having only one version of the gene, they're going to pass on one of those copies of that one version of the gene to the offspring. And in this case, you end up with all plants receiving a big P from the purple parent, and all of the white ones, them all receiving a white allele from the white parents. And when we draw out what happens crossing that F1 generation, we find in 3/4 of the offspring, they have at least one copy of that big P allele, which means they're going to be purple. But in 1/4 of the offspring, they're going to get small p's from both parents, which means they will appear white. So this is Reginald Punnett. He was an early geneticist, active in the late 19th and early 20th centuries. He was married to a woman named Evelyn Maud Froude. And they didn't have any children, but we can imagine that if they did have children, we could make predictions about their children by drawing a Punnett square. So here's a Punnett square of Punnett. Here he is. He's got brown eyes, and if he himself had a brown-eyed parent and a blue-eyed parent, he could pass on either that brown eye allele or the blue eye allele to his children. Evelyn here, this isn't really Evelyn, but it's another old-timey picture of someone who could be Evelyn, has blue eyes, so she can pass on only the blue-eyed allele, which is recessive to brown, to her offspring. So when we draw out the Punnett square showing the genotypes of the potential gametes from both parents, so dad's gametes and mom's gametes, we can make an estimate of how likely it is that their children will have either brown eyes or blue eyes. So there's a 50% chance that their children would have brown eyes and a 50% chance that their children would have blue eyes. So maybe next time you see someone who you're attracted to, you might be able to come up with a Punnett square in your mind about what the likelihood of your children having a certain eye color is. So again, if you've got two copies of the same allele for a gene, we say that you are homozygous. If you have two different alleles, two different versions of the same gene, we would say you are heterozygous. So homozygotes are true breeding, heterozygotes are not. So sometimes we want to try and figure out what a genotype of an organism is, but we can't necessarily tell because the dominant phenotype might be masking the presence of a recessive allele. So we need to figure out how to distinguish between the appearance of an organism, its phenotype, and the genetic basis for that appearance, which is the genotype. So a question you might have is, how do you figure out what your genotype is if you've got the dominant phenotype? There's a 1/3 chance in purple plants that you're going to have a homozygous individual, and there's a 2/3 chance that it's going to be heterozygous in the purple plants. So how do you know whether it's one or the other? You know because you can do what's called a back cross. So you take a purple flowered plant and cross it with a white flowered plant, and you see what the progeny are that develop. So how that would work is you have a purple flowered plant, and you want to know, is it homozygous dominant or is it heterozygous? So you can do a test cross with a known recessive white flowered plant, and if it is PP, big P, just like the first experiment, you expect to see all purple flowered plants. However, if your purple flower plant is heterozygous, it means it's about equally likely that you're going to get purple flowered offspring or white flowered offspring. So all of that Mendel deduced looking at a single character, flower color. But he came up with another hypothesis based on that particular set of experiments, which is, do the genes get distributed in packets, or is it just kind of a molecular free-for-all?